Examined under a microscope, every living organism is made up of billions of cells. Sometimes, within those, you will find bacteria living. This bacteria is called Wolbachia, and what makes it interesting is the close relationship it shares with its hosts and the many different strains found around the world. After years of research, scientists have found a way to use Wolbachia as a control method for the deadliest animal on Earth, the mosquito. It is estimated that Wolbachia infects the cells of nearly 70% of the world's insects. Each species is infected with a different variation or strain. The success of Wolbachia is due to its ability to manipulate the reproductive process of its hosts. It has evolved a number of ways to ensure that it gets passed along from one generation to the next.
Human mortality and morbidity worldwide are being affected by vector-borne viruses like dengue, chickpea, and malaria, with dengue being the most significant arboviral disease in tropical and subtropical communities. Specifically, the yellow fever mosquito is considered the primary vector of two of these viruses, dengue and chickpea. Controlling the mosquito vectors is the only way to prevent human cases of these diseases due to the fact that there are no effective vaccines. Introducing Wolbachia pipianus, a life-shortening strain, into the mosquito population shortens their adult lifespan without eliminating the whole population. This study looks into how the W. melpop CLA-infected mosquitoes may have altered the vector competence for arboviruses due to the W. melpop strain providing protection against RNA viruses in Drosophila.
For this experiment, 5 different lines of Aedes aegypti mosquitoes were used. The Aedes aegypti mosquitoes, commonly known as yellow fever mosquitoes, were bred and collected from Cairns, Australia. The Wolbachia strain used in this study was a derived W. melpop strain from infected Drosophila flies known as W. melpop CLA. The effect of Wolbachia on vector competence was tested in 2 mosquito genetic backgrounds, the inbred PGY-P1 line and a genetically diverse line derived from this original inbred PGY-P1 line known as PGY-P1 out. In order to accurately test the effectiveness of the Wolbachia-infected mosquitoes, these 2 mosquito strains were also compared to their tetracycline-treated counterparts, known as PGY-P1 tet and PGY-P1 out tet, that lacked the Wolbachia infection. A wild-type strain of Aedes aegypti was also used to serve as a negative control. Prior to exposure to the experimental conditions, these mosquitoes were kept in a controlled environment insectary. The dengue virus and chikungunya virus were used to examine possible interactions with the Wolbachia-infected mosquitoes. 3 to 5 day-old female mosquitoes were used for the dengue infection experiments, and 7-day-old females were used for the chikungunya experiments. Mosquitoes were starved for 24 hours prior to exposure to the dengue or chikungunya virus. These mosquitoes were then fed an artificial blood meal spiked with either the dengue or chikungunya virus. In addition to orally administering the dengue virus through blood virus mixtures, mosquitoes were also intrathoracically injected with the dengue virus. Titration of the dengue and chikungunya virus stocks in blood virus mixtures was performed using cell culture enzyme immunoassays. For the dengue virus, C636 cell monolayers were inoculated with virus dilutions and incubated. For the chikungunya virus, titrations were performed in viro cells and then incubated. In addition to exposure to the dengue virus and chikungunya virus, the Aedes aegypti mosquitoes, as well as another mosquito species, the Aedes fluviatilis, were also infected with the protozoan parasite Plasmodium gallinaceum. Plasmodium gallinaceum which causes malaria in poultry, was administered to 3 to 5 day-old female mosquitoes by feeding from infected chickens. DNA was extracted 15 days after infection in mosquitoes. Plasmodium gallinaceum and the dengue virus were detected by quantitative polymerase chain reaction, and the chikungunya virus was detected through reverse transcription polymerase chain reaction. It was also necessary to test whether resistance to the Wolbachia was related to a change in the innate immune system. In order to account for this possible confounding variable, a selection of immune genes from 5 to 6 day-old female PGY-P1 out and PGY-P1 out TET mosquitoes was analyzed by reverse transcription polymerase quantitative chain reaction.
The following figure shows the results of quantitative PCR analysis looking for dengue virus in mosquitoes. The top half showing genomic and the bottom half showing antigenomic. As you can see, for the PGY-P1 type, when there was Wolbachia inside the mosquito, there was no presence of dengue until 14 days post-infection, which even then there was very small amount of dengue. And then for the PGY-P1 out type, again, with the presence of Wolbachia, there was little to almost no presence of dengue within the mosquito. The following pictures show the results of immunofluorescent staining of mosquitoes sections to see localization of dengue virus, which is in red, and the Wolbachia, which is in green. And as you can see, when the pictures are contrasted with each other, whenever there is Wolbachia present, there is no presence of dengue. The following figures and images show the results of Aedes aegypti and Aedes fluviatilis mosquitoes feeding on Plasmodium gallinaceum infected chickens. Figure A shows the intensity of oocytes within these different mosquitoes. Figure B shows the midgut staining of the mosquitoes, and figure C shows the relative abundance of the Plasmodium gallinaceum with the use of quantitative PCR analysis. And what you can gather from all this is that Fluvitalis had the most Plasmodium gallinaceum, then the Aedes aegypti without Wolbachia had the second to highest, and then the Aedes aegypti with Wolbachia had the lowest amount of the Plasmodium gallinaceum present.
Expression levels of 11 immune genes in the wild-type MelPop CLA infected and uninfected control line were compared using two different cohorts of mosquitoes. Graphs A and C represent cohort 1, while graphs B and D represent cohort 2. Both cohorts showed an upregulation in cecropin, defensin, thioester-containing proteins, and C-type lectins, while in the presence of wild-type MelPop CLA. These genes are responsible for encoding immune effector molecules. Fibrinogen-related protein 18 levels remain the same in cohort 2. There was a significant increase in IMD and JAK-STAT signaling pathways. This was a difference between the two cohorts. This suggests variation between the cohorts were greater than the changes induced by Wolbachia. The effector genes that were affected by Wolbachia increased by as much as 100 times the normal regulation.
One of the mechanisms proposed for the reduction of viral infections with the Wolbachia is the competition with the viruses for key components from the host. None of the players in this system can synthesize cholesterol on their own. The mosquitoes get it from their diet, while the Wolbachia and the viruses get it from their hosts. This creates a competition between the Wolbachia and the viruses for the limiting nutrient, the cholesterol. The importance of other interferences and competition for other nutrients have not yet been studied.
The results from the experiment showed that the wild-type MelPop CLA Wolbachia strain reduced the ability of the arboviruses and malaria parasites from infecting the mosquito. some of the data suggested there was an upregulation of immune effector genes. It is believed that this upregulation is due to the presence of the wild-type MELPOP CLA strain. However, the currently accepted signaling pathway is not affected by the Wolbachia. Therefore, the mechanism that prevents the infection is still somewhat unclear.